So, good morning or good afternoon, everybody. It's a tremendous pleasure and an honor to continue our Global IMMUNOTALK series with another highly distinct speaker. Today it's Eui-Cheol Shin. I hope I pronounced this somewhat correctly. I was rehearsing a little bit before. And it's really a pleasure to welcome you. And one thing I should really bring up is that for us, it's morning or evening. For you, it's in the middle of the night. So it's 1 a.m. You're joining us from Korea. So thanks for doing that. We highly appreciate it. Some speakers have pre-recorded the meetings. You decided to do this live, which is really great. And thanks for that. So I'd like to say a few words about your CV and background. So you are a professor at the Graduate School of Medical Sciences and Engineering at the Korean Advanced Institute of Science and Technology, where you told me, which is the Korean equivalent of the MIT. You're also director of the Center of Viral Immunology at the Korean Virus Research Institute and Institute for Basic Sciences. Both of this is in Daejeon in Korea. You started your career with graduating from medical school, and it was at the Yonsei University College of Medicine in Seoul in Korea. Afterwards, you quickly transitioned to obtain a PhD in microbiology and immunology from the same institution. Afterwards, you were a postdoctoral fellow with Barbara Rehrman, if I remember correctly. And that was at the NIH and specifically at the National Institute of Diabetes, Digestive and Kidney Diseases of the NIH. And afterwards, you returned to Korea and you became progressively, or you progressed from assistant professor to associate professor. Now I assume you're a full professor. And you're heading a laboratory of immunology and infectious diseases still at Daejeon in Korea. You're also an adjunct professor at the University College of Medicine of Yonsei University. And you're a member of the Korean Academy of Sciences, member of the National Academy of Medicine of Korea. And your research interests are mainly in viral immunology, viral hepatitis, tumor immunology, immune aging, human immunology, and in particular, you have lately been focusing on T-cell-mediated immune pathogenesis, senescence of T cells, reinvigoration of exhausted T cells, and immune responses to SARS-CoV-2 and the disease pathogenesis of COVID-19 infections. And now I can continue with this long, and I could highlight your astonishing list of papers and prior achievements and grants, but I will cut my introduction usually very short. You are the speaker today, so I'd like to thank you very much again for joining us. And I will start with our usual opening question, which is this time, I know you will be talking about bystander T cells, and my question is, where do you see actually the key challenges and the main sort of things that still need to be discovered in the area of the biology of bystander CD8 T cells? So thanks for joining us.
So actually, the bystander T cell activation is kind of abandoned area or ignored area in the history of immunology. I think so. So in my talk, I'm explain about that in my talk. But now, currently, most important question in this field. So we don't know yet the role of the bystander activated CD8 T cell in human disease. So one of my hypotheses, they might contribute to the pathogenesis of some chronic inflammatory disease. So no one just demonstrated precisely this mechanism. So now I'm trying to do that currently in my lab. Yeah.
Very good. I can only agree with this because I think this is an area we have all underestimated, and I think it has gone through a huge revival over recent years. And I think we're all very happy to now hear more about this. And thank you very much again.
Thank you. So now, can I start now the presentation now? Yeah. Let me share the slide. Then from now, I'm going to talk about the regulation of cytokine-induced bystander CD8 T cell activation. So as you know very well, during viral infection, the viral antigen-specific CD8 T cell will be activated. So when I was postdoctoral fellow in NIH, I studied the HCV-specific CD8 T cell response by using the HCV-infected chimpanzee. So with this expertise, so during the COVID-19 pandemic, I also studied the SARS-CoV-2 specific CD8 T cell in patients with COVID-19 or individual immunized with COVID-19 vaccine. So during COVID-19 pandemic, so in my lab, we successfully demonstrate that the SARS-CoV-2 specific CD8 T cell are not exhausted but functional. Also, we also successfully showed that the successful generation of the stem cell-like memory T cell after infection or after vaccination. In addition, at the year of the Omicron variant, we also examined the variant-specific CD8 T cell response after vaccination or after breakthrough infection. But so in my talk, I'm not going to talk about the antigen-specific CD8 T cell. So today, I'm going to talk about antigen non-specific. In other words, TCR-independent activation of bystander memory CD8 T cell. So the bystander memory CD8 T cells activation was firstly reported by the Jonathan Sprant group in 1996. In this paper, so the Sprant group, they showed that by using mouse model, infection model, antigen non-specific bystander CD8 T cell can proliferate during viral infection. In the next year, in 1997, the Dr. Zinkonagel's lab, they repeated the Sprant group's experiment. And then they conclude that in next year, 1997, the bystander activation is true, it happened. But in terms of quantity, it is not so important. So now I'm just citing this sentence from their abstract, and they conclude that in quantitative conservation suggests that this is usually not a major biological consequence. So after this Zinkonagel's paper, the bystander activation of the CD8 T cell, it was not so popular area. So, but time to time, in human study, the bystander activation have been demonstrated in certain viral infection, including HCV, HBV, EBV, HIV, hantavirus, and influenza virus. Also in mouse model, the bystander activation CD8 T cell was reported in Listeria monocytogenesis infection. In this case, bystander activated CD8 T cell contribute to the early control pathogen. But in other models, for example, Leishmania major model, infection model, this bystander activated CD8 T cell, it contributes to the increased immunopathology. So it's depending on the infection model, this bystander activation in mouse model, it shows some beneficial effect or detrimental effect to the host. So in this way, the clinical significance of bystander CD8 T cell activation was not so clear in the past time. But recently in my lab, around 6 years ago, we successfully showed that this bystander activation plays an important role in HIV infection, Hepatitis-A virus infection. In the first part of my talk, I'm going to show you our previous published paper. So what we discovered about the bystander activation in HIV infected patients. So before explaining about that, let me show you a little bit briefly explain about what HIV is. From the past 20 years in South Korea, the HIV infection have several some outbreak. So during this outbreak in my lab, we could obtain the patient sample. Then we could have chance to analyze the T cell response in this HIV infected patient. But the HIV infection is very unique disease in terms of the host damage. The HIV infection is usually asymptomatic in children, but only it induces the liver injury only in the patient. And this graph is showing natural course of HIV infection. And after HIV infection, there's 4 to 6 weeks incubation period. This is not days, the weeks. So, but during this 4 to 6 weeks incubation period, virus will rapidly replicate, and even virus will be secreted to the feces. in that way, so it can be transmitted to other people. But during this incubation period, they are asymptomatic, even in adult patients. But after this incubation period, the ALT abruptly increased. ALT, as you know very well, the liver enzyme. So hepatitis, liver injury, it begins in the 4 to 6 weeks later than this the infection. Then at the time of this the ALT peak, the patient will experience a symptom like a fever, more malaise, more general malaise, and so jaundice, more like that. And this the ALT peak time point, the patient will develop HIV-specific antibody and HIV-specific T-cell response. So, but then why? In adult patient, the HIV infection result in this the ALT peak, liver injury. So it was known that in 1980, some several German group, they showed that the liver in the HIV cell, although it replicates very rapidly in hepatocyte, they don't kill the virus itself. It doesn't kill the hepatocyte. So we cannot explain this the liver injury by direct cytopathic effect of virus. Instead of that, the German scientists around about 30 years ago, or more than 30 years ago, they explained that the liver injury is caused by HIV-specific CD8 T cell by their cytotoxic function. So in other words, they conclude that the HIV-specific CD8 T cell response is optimal, then it will be beneficial for the viral clearance in the patient. But the T cell response is too much excessive, exaggerated, then the patient will experience the severe liver injury. But what's the optimum, or what's the excessive, exaggerated response? So no one knows about this, what's the optimal level of the T cell response. So with this background, so during the outbreak of the HIV infection in South Korea, we obtained patient PBMC, the whole blood sample, and we obtained the PBMC. When at the beginning, we simply analyzed the activation status of CD8 T cell. So now I'm showing you the CD38 and HLA-DR double positivity of CD8 T cell. The CD38 and HLA-DR double positive cell means that the activated CD8 T cell. The compared to healthy donor, in HIV-infected patient, they have significantly increased the activation of CD8 T cell. And the degree of activation nicely correlates with the serum ALT level. And such kind of correlation was also observed when we examine Ki-67 proliferation marker. The significant increase in patient and nice correlation with the serum ALT level. So after getting this data, so my lab, initially, we just thought that, oh, maybe the known knowledge, so it is true. So already I told you that the exaggerated CD8 T cell response, it might contribute to the liver injury. So the more activation and severe liver injury, more severe liver injury, and more proliferation, more severe the liver injury. But the graduate student who was the first author of this paper, the Dr. Jie Kim, she pointed out that something weird. So what's weird? So in certain patient, the ALT level is very high. Oh, yes, more 9,000, more like that. But in such patient, nearly 90%, more than 90% of total CD8 T cell are activated. More than 90% of the CD8 T cell are proliferating during acute HIV infection. So we cannot say that 90% of total CD8 T cell are HIV, single virus specific. So we could not imagine that. So we have thought that something else happened in this HIV infection. So then, what will be how we can explain about the 90% of total CD8 T cell are activated during HIV infection? So after getting the notion, we decide to examine the antigen specificity of this activated CD8 T cell. To do that, we applied the HAV-specific MH class 1 tetramer. And now, so I'm showing you the tetramer positive cell in HAV-specific tetramer from the representative patient. And after gating of this the tetramer positive cell, we examined 38 and HADR. And more than 90% of this HAV-specific T cell, CD8 T cell, are highly activated. Then how about the HAV unrelated virus specific CD8 T cell in this patient? In this case, we use CMV, EBV, influenza A virus tetramer. After successful detection of this tetramer positive cell, we gated this tetramer positive cell, and then we examined 38 and DR. As you can see here, 17%, 78%, 76% are 38 and DR double positive. But then, how about the CMV, EBV, influenza A virus specific CD8 T cells activation status in healthy donor? In healthy donor, this tetramer shows 0%, 5%, 4%, 38 HADR double positivity. So now I'm showing you the summary data from the multiple patient. In the HIV infected patient, HIV specific CD8 T cell, nearly 100% of them are highly activated. But in the case of CMV, EBV, flu specific CD8 T cell, not 100%, but considerable percentage of those CMV, EBV, flu. It means that bystander, the memory CD8 T cell, are activated. This 38 DR double positivity of this bystander CD8 T cell in healthy donor is just less than 5%, as you can see here. So it means that this the simple analysis, tetramer analysis, we successfully demonstrate that not only HAV specific CD8 T cell, but also bystander memory CD8 T cell are activated during HIV infection. So now I'm showing you another piece of evidence of bystander activation, not by 38 and DR. In this case, I'm showing you Chi67 and granzyme B for not only HIV spec CD8 T cell, but also EBV and CMV spec CD8 T cell patient. In the previous slide, I showed you the evidence of bystander activation by using peripheral blood mononuclear cell. Then how about in the liver? So during the outbreak in South Korea, we could obtain the explanted liver during liver transplantation. The certain patient has fulminant hepatitis, so they got the emergency, the liver transplantation. Then we could obtain the explanted liver, HIV infected liver, and then we apply the tetramer again. As you can see here in the HIV infected liver, HIV specific CD8 T cell are 98% are activated, and CMV spec CD8 T cell, 94% of them are 38 and DR double positive. So in this way, we found that HIV unrelated virus specific CD8 T cell are activated in patients with acute HIV infection. So not only in the peripheral blood, but also in the liver. So now I found that there are two different types of activation in HIV infected patients. The conventional activation, it means HIV specific activation, and unconventional bystander activation. And then what's the clinical relevance of these two different types of activation? So to answer this question, we simply perform the simple correlation analysis. In this correlation analysis, we quantify the number of each virus specific CD38 and HADR double positive CD8 T cell per microliter of blood. In the case of the flu, EBV, CMV specific CD8 T cell. Each virus, bystander, the specific CD8 T cell, bystander, the memory CD8 T cell, their activation, the number of activated CD8 T cell are nicely correlated with serum ALT level. So it means that it indicates that so maybe the bystander activation might contribute to the liver injury. But in the case of HIV, the tetramer positive cell, the HIV tetramer positive and 38 HADR double positive cell, their number, instead of the positive correlation, they show the inverse correlation with the serum ALT level. So with this the inverse correlation, we could not say that maybe this the HAV tetramer-positive cell they contribute to the liver injury. We could not say that. How about the kinetic analysis? So usually this HIV infected patient, they admitted to the patient and then they are hospitalized. Then 1 to 2 weeks later, they're discharged. And meanwhile, there's no specific treatment and just observation. So the careful observation during the hospitalization. And then it takes from the admission to discharge, it takes 1 to 2 weeks. Then meantime, serum ALT significantly decreased. And then the double positivity among the total CD8 T cell or double positivity among bystander CD8 T cell, this percentage significantly decreased at the time of discharge. But the double positive cell, the frequency among HIV-specific CD8 T cell, it did not decrease at the time of discharge. Even with this the kinetic analysis, these data indicate that bystander CD8 T cell activation nicely correlate with the liver injury during acute HIV infection. So it indicate that maybe this the liver injury might be caused by bystander CD8 T cell. Then how about the role of HIV-specific CD8 T cell? So now I'm showing you again this HIV, the specific CD8 T cell and serum ALT level inverse correlation again. Then we investigate HIV-specific CD8 T cell response in different way. So we perform ELISpot assay to measure the HIV-specific CD8 T cell response in different way by using the overlapping peptide or by using A2 restricted, the HIV epitope, the mix. Then again, with this ELISpot assay, we could find the inverse correlation between the HIV-specific T cell response and serum ALT level. Then, how about the relationship between the HIV-specific T cell response with the serum viral titer? There's again inverse correlation. It means that if patient have higher T cell, stronger T cell response, then this patient will have lower viral titer and low serum ALT level. But such kind of the relationship with the viral titer, it was not observed by the total CD8 T cell activation. So it means that in very brief summary, I can say that HIV-specific CD8 T cell show inverse correlation with the viral titer. It means that this the conventional activation, it contributes to the viral control. But bystander CD8 T cell, it contributes to the host injury. So now with this simple analysis, we could conclude that there are two different types of activation, have two different types of the role during the HIV infection. Then what induces the bystander activation? We hypothesized that maybe common gamma chain cytokine may play a role. So we found that IL-15 is increased in the serum and also in the liver. Also, we found that the IL-15, we could successfully stain this IL-15 in the HIV infected liver. Then, from this point, we recapitulated the bystander activation by using in vitro simulation of the IL-15 into the healthy donor-derived memory CD8 T cell. In this experiment, we treated healthy donors PBMC using the IL-15. Then we found that CMV flu-specific CD8 T cell activated by IL-15 without TCR engagement. And also, we performed the NK-like killing activity of the IL-15 treated healthy donor CD8 T cell or the CD8 T cell sorted from HIV infected liver. So in this figure, the high purely sorted CD8 T cell, memory CD8 T cell from healthy donor, and we treated them with IL-15, then perform the NK-like killing activity by using K562 as a target cell. And we found that this IL-15 treated memory CD8 T cell can kill the K562. So this one was surprising data because K562, they do not express the MH class 1. So this the killing activity cannot be explained by TCR-MH interaction. So such kind of the NK-like activity, killing activity could be seen. So when we sorted the CD8 T cell from HIV-infected liver. And also, we perform the similar kind of assay by sorting CMV-specific MH multimer positive cell from peripheral blood from HIV-infected patient. Even after sorting the CMV-specific multimer from peripheral blood of the patient, they could kill the K562. So in this way, we found that there's the innate-like cytotoxic activity, NK-like cytotoxic activity. Then which receptor mediate this NK-like innate-like cytotoxicity? To answer this question, we treated healthy donors memory CD8 T cell with IL-15. Then we examined activating NK receptor expression. And we found that NKG2D and NKP30 are regulated by IL-15 stimulation. And then we perform again this K562 killing assay by using CD8 T cell from the HIV-infected liver and or IL-15 treated CD8 T cell by using blocking antibody against NKG2D or NKP30. As you can see here, NKG2D blocking antibody or NKP30 blocking antibody successfully abrogate the NK-like killing activity. It means that the NKG2D and NKP30 mediate the NK-like cytotoxicity of IL-15 stimulated the bystander activated CD8 T cell. Then finally, in this the previous literature publication, we show that this killing activity again have a nice correlation with the liver injury. So even when we the purely salt CD8 T cell and perform K562, the target cell killing activity, then the certain patient showed a higher innate-like cytotoxicity, even from the peripheral blood CD8 T cell. So with this value, we perform the correlation analysis with the serum ALT level. There's very nice correlation between the peripheral blood CD8 T cells, the NK-like killing activity, with the serum ALT level. So 6 years ago, we successfully showed that in HIV infection, the bystander activation can happen by IL-15. And the IL-15-induced bystander activated CD8 T cell can exert innate-like cytotoxicity by NKG2D or NKP30. So this is the summary of our previous publication. Oh, now I'm showing you again this graphical abstract of the previous publication. So the IL-15 will be produced from HIV-infected hepatocyte and also other myeloid cell. And this IL-15 will activate the pre-existing memory CD8 T cell without TCR engagement only by cytokine, bystander manner. And this bystander activated CD8 T cell will upregulate NKG2 and NKP30. And then with this NKG2 and NKP30, they can kill the target cell. In this way, liver injury will be caused by bystander activation. After this 6 years ago, our publication, so we continued the bystander study. So meanwhile, we successfully showed that IL-15 upregulates not only NKG2D but also CCR5, and the bystander activated CD8 T cell can migrate to the infected liver by CCL5 and CCR5 interaction. And also, we found that CD5 molecule can suppress this IL-15-induced activation of human memory CD8 T cell. In addition, we also published a paper that IL-15 can be produced by hepatocyte, epithelial cell, including hepatocyte, by type 1 or type 2 interferon. But in this case, not only IL-15, but also IL-15 receptor alpha can be produced. In this way, even if not only myeloid cell, but also epithelial cell can do the IL-15 transpresentation to NK cell or memory CD8 T cell. So this one, so in my knowledge, this HIV infection, for the first time, we successfully showed that in human disease, among human disease, the bystander CD8 T cell can do some role, pathological role. But after our publication by other group, the similar the report was published. So one Taiwanese group, they performed a similar analysis in the chronic Hep C patient, HCV infected patient, and they found that the similar mechanism operate in chronic HCV infection. And also, so in my lab, with the collaboration with the Dr. Su-Young Park's group in my same department, we found that in alopecia areata model, so this is not viral disease, so like chronic inflammatory disease, in alopecia areata model, the similar mechanism, it play a role in the pathogenesis of alopecia areata. In addition, there are some recently, there are more and more paper showing the similar mechanism in viral disease, other viral disease. For example, in human HBV infection, one meta-archive paper reported the same similar mechanism, and even mouse Zika model also showed the same kind of the bystander activation. Then, from now, I'm going to show you the some unpublished data. But then before talking about this, our unpublished data, I'm going to show you this very unique feature of NKG2D. So in our previous publication, we found that NKG2D is upregulated by IL-15 stimulation, but not by anti-CD stimulation. But we found that surprisingly, when you combine anti-CD3 with IL-15, so there's the down modulation of this NKG2. So it means that the anti-CD3 T-cell stimulation counteract this IL-15-induced NKG2D upregulation. So in that time, we didn't know the mechanism, why TCR signal counteract. But so even without the mechanism, we use the data for supporting the bystander activation in HIV-infected patients. So in this data, in this graph, I'm showing that in healthy donor, in memory cell or CMV-specific T cell, NKG2 is not so highly upregulated. But in HIV-infected liver and the patient, HIV tetrapositive cell, there's no upregulation of NKG2D, but only CMV and RSV-specific T cell, there were NKG2D upregulation. So why? In HIV-specific T cell, in this case, the T cell will receive not only IL-15 stimulation, but also TCR stimulation. So the TCR counteract this NKG2D upregulation. But in CMV and RSV-specific T cell, there's no TCR stimulation, so NKG2D upregulated. So with this data, we describe that NKG2D upregulation on human memory state T cell is an indicator of activation by IL-15 in the absence of T cell stimulation. We said that this can be a marker indicator. But in this one, is the knowledge only applied to human CD8 T cell. So also we found that in mouse model, NKG2D is upregulated not only by IL-15, but also by anti-CD3. In the mouse model, the molecule like IFTM3 can be a marker for the bystander activation. But then, with this background, in my lab, the postdoctoral fellow, the Ho-Young Lim, he asked a question, then why? By which mechanism the TCR counteract this IL-15-induced NKG upregulation? So to answer this question, he repeated the previous experiment. So in this data, again, we confirm that IL-15 upregulates NKG2D, but TCR cannot. But when you combine both signals, the stimulation, TCR stimulation counteract the IL-15-induced NKG2D upregulation. But this kind of counteraction can be seen only in NKG2D. Other activation marker or proliferation marker, there's additive or synergism effect between IL-15 and TCR stimulation. So the Ho-Young, he concluded that TCR stimulation counteract so only certain aspect of IL-15-induced activation of memory CD8 T cell, including NKG2 regulation. So in next step, Ho-Young performed bulk RNA analysis. In this case, we used the healthy donor CD8 T cell, memory CD8 T cell from 10 donor, and then we treated this the salted CD8 T cell with IL-15 or CD3 antibody or combination, and we performed bulk RNA analysis. And as you can see here, many more genes show the synergism or additive effect that can be explained. These genes are upregulated by IL-15, also upregulated by anti-CD3 antibody and synergism. But now you can see this the cluster 4, for example, expressed by upregulated by IL-15, but not by CD3 antibody. But when you combine this the IL-15 and CD3 antibody, there's no upregulation. So now I'm showing you such kind of the very interesting gene, the gene set, this one, upregulation by IL-15, and but counteracted by anti-CD3 stimulation. We found that such kind of IL-15 specific gene set are highly enriched with this the interferon pathway and NK cytotoxic pathway. And this the opposite one. It means that this gene cannot be upregulated by IL-15, but only by CD3. These genes are highly enriched with this NFAS signal, the pathway gene. And we could confirm this the interferon, the pathway gene and NK cytotoxic gene are highly enriched in this the IL-15 spec gene set by GSEM. Then Ho-Young asked the next question: why? What's the mechanism? And then simply Ho-Young hypothesized that maybe some signaling, this the basic information about the IL-15 signal and T cell signal will give us some hint, answer to us. And he found that this the calcium pathway, so only this one is only the present. This is present in TCR pathway, signal pathway, but not in IL-15. So he hypothesized that maybe this calcium-calcineurin pathway play a role for this counteraction. So in the next step, he performed again the bulk RNA analysis, but in this case, he did not use anti-CD3. He used ironized calcium iron 4 instead of anti-CD3 antibody. Now, as you can see here, IL-15 upregulated certain gene set, and then this gene could not be upregulated by inomycin, but by combination, it was not upregulated. So now I'm showing you again in this way, and this gene again, the interferon response and NK killing activity, killing the cytotoxicity pathway gene are highly enriched in this gene. And now this one is showing just in the single gene level, confirming this such data. So we found that calcium signals are involved in TCR-mediated inhibition of IL-15-induced NK-like activation of CD8 T cell. So we confirm this one again by FACS analysis. So when we stimulate the memory CD8 T cell with IL-15 NKG upregulated, inomycin, no upregulation, and by combination, we could suppress the NKG to the upregulation. And this one is very interesting data. In this case, we used the IL-15 and anti-CD3, the combination. By this combination, there's not so much NKG to the upregulation. But when we suppress the calcineurin pathway by tacrolimus, then by suppressing calcium-calcineurin pathway, this NKG to the level is paradoxically increased. So why I'm saying the paradoxically? Because the tacrolimus immunosuppressant can upregulate NKG to D in this very specific circumstance. And now we confirm again such kind of data by using the PBMC acquired from the acute HIV infected patient. And again, this one is also the killing activity. So if I stimulate the memory CD8 T cell with IL-15, then this the IL-15 stimulated memory CD8 T cell can kill K562. But when I combine this IL-15 with the inomycin, then there's no NK-like killing activity. Then so far, I showed that the TCR independent IL-15 stimulation in this such kind of the situation, NKG2 will be upregulated, and this cell can exert NK-like cytotoxicity. And when you combine IL-15 with TCR, then TCR stimulation by calcium signaling pathway will suppress such kind of bystander activation. Then when we add, in the presence of IL-15 and TCR stimulation, when we add calcineurin inhibitor, then this calcineurin inhibitor can suppress the TCR's mediated counteraction. In that way, again, NKG2D will be upregulated by calcineurin inhibitor. And then NK-like cytotoxicity will be also enhanced. Then, what's the more deeper molecular mechanism? Then now, currently, Ho-Young is trying to answer this question. And now I'm showing you our one of the preliminary data. And then according to our preliminary data, Ho-Young showed that the IL-15 induced this NKG2D regulation, it depends on AP1. But in TCR stimulation, NFOT will be activated by calcium-calcium pathway, but NFOT-C1 can make a dimer with AP1. Then in this way, AP1 will be occupied by NFOT-C1. Then AP1 cannot do any more this NK regulation. This is our current working hypothesis. And this hypothesis is now recently it was demonstrated by a very simple experiment. So we found that the NFOT-C1, the previous more than 25 years ago, very old publication, they found that which amino acid motif is important for the dimerization between NFOT-C1 and AP1. So this the mutant cannot make a dimer with AP1. So with this the wild type and this mutant, we perform the transfection analysis. In this case, we transfected CD8 T cell, memory CD8 T cell with the wild type NFOT-C1 and mutant. This mutant cannot dimerize the AP1. Then that's the increase of the NFOT-C1, the expression level by transfection. And then with the IL-15, the stimulation, the NFOT-C1 wild type can suppress NKG2D upregulation. But the mutant cannot suppress this NKG2D, the expression. In that way, for the TCR mediated the counteraction on NKG2D regulation, it can be explained by the sequestration model between AP1 and NFOT-C1, by dimer formation. Then from now, I'm going to show you very briefly the decode data. So decode data for what? So I showed you the slide before. So now we focus on this gene set. You may remember that this gene set can be upregulated only by IL-15, but not by the co-presence of IL-15 and anti-CD8 stimulation. So we define that this gene set is a bystander activation gene set. By using this gene set, we can find another disease, so in which bystander activation play a role for some pathogenesis. So to validate this bystander activation gene set, we perform the decode analysis using the HIV-infected patient peripheral blood. In this case, we perform single-cell RNA analysis by using DNA barcoded MH class 1 multimer. In this multimer analysis, we use not only HIV multimer, but also CMV, EBV, flu multimer. In this way, in HIV-infected patient, we could detect not only HIV-specific CD8 T cell, but also bystander CD8 T cell. So this one is important data. Compared to HIV-specific CD8 T cell, this is the bystander, the memory CD8 T cell. highly enriched. They have the higher enrichment score for this bystander activation genes. The GSCA confirmed this the gene enrichment. It means that really this the IL-15 activation specific gene set, bystander activation gene set, are really the upregulated in the bystander, the memory CD8 T cell in HIV infected patient. So in this case, we used the anti-CD3 stimulation specific gene set, mainly NPAT signal pathway gene set, which one is the upregulated by in HIV infected the specific CD8 T cell compared to bystander CD8 T cell. So this the data is showing this the same data in single gene level. So we conclude that bystander activation gene set is enriched in bystander CD8 T cell, but not in HIV specific CD8 T cell. And also, we confirm this the bystander activation gene set validate again in mouse model of bystander activation. In this the mouse model of bystander activation, basically we use flu, but we using OT1 memory cell as a bystander pre-existing memory cell. In this case, already we published this data in 2 years ago, and we used this gene set and the volcanistic data, and we confirmed that in the bystander activation gene set is highly enriched in the situation of bystander activation. So actually in this the mouse model, bystander activation can occur by the PLA, the flu infection, but in the case of TCR activation, so not only the combined activation, not only PLA, but also synthetic peptide also delivered for the T-cell stimulation. So only this bystander gene set was observed highly enriched in the bystander activation stimulation. So in this way, we confirm this is our gene set in mouse model. In several slides, I'm going to show you the relationship between aging and bystander activation. At the beginning of my talk, I explained about that HIV infection are usually asymptomatic in children, but is induced very severe liver injury in adult patients. And now, so with this, the bystander activation theory can explain why there's some age dependency in terms of the severe liver injury. So as you can imagine very easily, the children have the small size of memory T cell pool. But during aging, this memory T cell pool will be enlarged. And even with the same amount of IL-15, the size of bystander activation is much bigger in this the adult or even elderly aged people. Considering this the point, maybe when we set up the study bystander activation, maybe we might use, it's much better to use dirty mouse or wilding mouse instead of the laboratory mouse, because it was already known that the memory pool size, T cell pool size, very small in laboratory mouse. In this aspect, now I'm going to show you some more data. In memory T cell population, especially in human, CD28 negative, 57 positive, this cell is known as replicative senescence memory CD8 T cell. And we found that, so when we examine the IL-15-induced activation between CD57 negative young memory cell and CD57 positive old memory cell, this IL-15-induced activation is much more exaggerated in aged, the replicative senescent memory CD8 T cell. Not by this just activation, but also in NKG2D upregulation is much more exaggerated in senescent CD8 T cell. And also NK lysozyme toxicity also same. So it means that senescent CD8 T cell are more hyper-responsive to IL-15 stimulation. And so far, I use this term bystander activation. But now in my lab, now I'm expanding our research not only in the conventional memory CD8 T cell, but also in unconventional CD8 T cell. And especially in human virtual memory CD8 T cell, we found that there are two different types of the virtual memory T cell in terms of marker. Among them, especially key-positive CD8 T cell are highly responsive, hyper-responsive to IL-15 stimulation. And also, in the liver environment, we identified very highly IL-15 responsive from small population by performing site-stick analysis. Especially this population express CD56 high, 161 negative, and NKG2 positive. Even they can kill the K562, this NK target, not only by NKG2D, but also by NKG2C. So let me summarize my talk. So in my talk, I showed you this bystander memory CD8 T cell they are activated in HIV patient, and they can contribute to the pathology, liver injury. And T cell stimulation counteract is the IL-15-induced NKG2 regulation by calcium pathway. So I can say that IL-15 and T cell signal they competitively regulate innate and adaptive activation of memory CD8 T cell. And senescent CD8 T cell are more responsive to IL-15, and even unconventional CD8 T cell, they can be activated without T cell stimulation only by IL-15. And so far in my talk, I focus on IL-15 in this bystander activation. But recently, I found that when I combine IL-15 with IL-12 and 18, there are more activation and more NK-like activity, more NK-like cytotoxic activity. So I think that this bystander activation is more complicated when you consider the several cytokine combination. In addition, so now I'm continuing my research in other disease to show that bystander activation plays some role, especially in the immunopathology in other disease. But at the same time, I found that acute HIV infection is a good model disease for studying immunopathology post-injury, resulting from bystander CD8 T cell activation. So HIV infection can be regarded as some prototype disease for bystander activation-induced immunopathology. So this work was done mainly by the Dr. Ho Young Lee in my research center, and my research is funded by the IBS KBRI, and also the initial stage it was supported by Samsung, the foundation. And thank you very much for your attention.
Well, thank you very much, Eui-Cheol, for this really great and very fascinating talk. I'm sure there's, as usual, many questions, but I'm waiting now for our slide. Here it is. As you all know, we are stopping the meeting here, and any questions to the speaker have to be addressed through the platform that was formerly known as Twitter. And with that, I'd like to close our meeting today. Thank you very much again for joining us. With this, we are finished, and I would like to say we look all forward to seeing you again next week when our speaker will be Osamu Takoishi. So have a great evening or nice remaining day to everybody. Bye-bye.